Hello. For those of you who don't know me, my name is Dr. Christopher Bolt, and I'm a second-year veterinary anatomic pathology resident at the University of Minnesota Veterinary Diagnostic Laboratory. Today, I'd like to virtually present to you a group of 3 feline cases that were submitted to the lab over approximately 2 years and ultimately diagnosed with a novel species of Neisseria pneumonia. The clinical history and clinical signs in all 3 cases was highly varied, but progressed in every case to marked lethargy and dyspnea prior to euthanasia or death. Abnormal thoracic radiographs were really the common antemortem finding. Here's an example of radiographs from case 3, and we can see multiple soft tissue opacities widely disseminated throughout the lungs, which was found in all 3 cases. Grossly, the lungs again were the main finding. So here, coinciding with those thoracic radiographs, we see numerous, multifocal to coalescing, beige to tan, relatively well demarcated, irregular to circular nodules, measuring approximately 0.5 centimeter to 5 centimeters in diameter, and affecting probably 50 to 80% of the lung lobes in all cases. Histologically, on hematoxylin and eosin staining, we can see areas of hypercellularity that coincide with those gross nodules, which, on higher magnification, we can see is an influx or an infiltrate of large numbers of cells into the alveolar spaces and alveoli. And at higher magnification, we can see that these are mainly made up of large numbers of neutrophils and smaller numbers of macrophages and variable amounts of fibrin. The alveolar septae, as well as the airways and vasculature, were exhibiting multifocal to coalescing necrosis, both coagulative and lytic. And there were central bacterial colonies, as seen in this slide, with approximately 1 micrometer basophilic coccobacteria. We did see bacterial emboli within vessels with surrounding vasculitis as well. And on gram stain, these bacteria were gram-negative and seen both extracellularly and intracellularly in macrophages and neutrophils. Samples of the lung in all 3 cases were submitted for bacteriology, and the cultures that grew were small, dull yellow, convex, and had a wrinkled appearance. Bruker-Maldi-Toff was run, and all of these colonies were consistent with N. animaloris, so Neisseria animaloris. 16S rRNA sequencing and full genome sequencing was run with in silico digital DNA hybridization, which found that all of these strains were 99.99% identical to each other, with the closest related strain being Neisseria animaloris at 46.6%, which fell below the threshold to be considered a novel species in these 3 cases. On the right, we can see our 16S RNA-based phylogeny, a phylogenetic tree, and our whole genome sequencing matched that. So feline pneumonia is actually a relatively uncommon disease, and Neisseria is a rarely reported cause of this disease. Since 1973, there's been sporadic cases, both in house cats and other feline species. There's really variable clinical signs and antemortem diagnostic testing results, but the most common result is that nodular thoracic radiographic appearance. Unfortunately, that's not pathognomonic. However, when you combine that with those gross and histologic findings that we described in these slides, that does lead to a relatively distinct appearance that we feel is indicative of Neisseria pneumonia when you see it. The diagnosis, unfortunately, is generally post-mortem, with only a single reported case of survival with antibiotic treatment. The pathogenesis, unfortunately, is still relatively unknown. So Neisseria species are common oral commensals in healthy dogs and cats based on previous studies. So it's hypothesized that there's translocation of this oral flora with distribution consistent with hematogenous spread, with the bacterial emboli that we found in our cases seemingly supporting this diagnosis and this hypothesis. The reason for the severe and sporadic nature of the disease is really not known, considering the commensal and common nature of these species in the oral cavity. So do the 3 cases that were submitted in these 2 years represent an incidental anomaly, or is this an emerging problem? We're not really sure. And is this novel species the cause of the majority of cases, or is that N. animaloris as reported in previous studies? Further molecular workup is really needed, including virulence genes, antimicrobial resistance genes, and looking into mutations to see whether this is a more pathogenic strain and whether this matches up with other strains in the past. So with that, I'd like to acknowledge all those who helped in this case, both care of the initial animals and diagnosis post-mortem. And here's references if you have any questions.